The goal of this procedure is to internalize and visualize biomolecules such as DNA or proteins labeled with organic fluorophores inside living microorganisms using a method based on electroporation. This is accomplished by first incubating electrocompetent cells with fluorescently labeled biomolecules, before transferring them into an electroporation cuvette. By applying a high voltage across the cuvette, transient pore formations on both the cell wall and cell membrane will enable biomolecular diffusion into the cells. Next, the cells are incubated in a rich medium to allow them to recover, before the excess non-internalized biomolecules are removed. Finally, the cells are transferred onto a low-fluorescence minimal nutrient agarose pad, and then a glass cover slip is overlaid on top to form a glass agarose sandwich. Ultimately, fluorescence microscopy is used to analyze the location, diffusion pattern, and dynamical properties of the labeled biomolecules inside the living cells.
We first had the idea for this method when we realized the limitations in live cell imaging due to the fast bleaching of photofluorescent proteins such as GFP. We reasoned that by loading living cells with biomolecules labeled with organic fluorophores, we would transplant in living microorganisms the main advantages of these fluorophores, that is, brightness, photostability, and small size. We then hope to follow the localization and diffusion of DNA or proteins in their native environments on much longer timescales, which would match many important biological processes.
The main advantage of these techniques over existing methods, such as microinjection, is that it can also be applied to microorganisms that are small.